Hello students, welcome back to yet another part in coordination in life processes. Before we further proceed in this part 2, let us quickly recap what you have studied in part 1. Feeling hungry, hunger pangs, taste and smell, tongue and palate, mouth, the munching machine. Let us know how different teeth in our mouth helps in mastication process. Students, let us do an activity 6 in order to know the significance of mastication with the help of teeth. Activity 6, observe the model or chart of jaw. How are teeth arranged? Are all teeth similar in shape and size? If you look at this picture, this is the left half of the upper jaw and this is the right half of the upper jaw. Similarly, this is the lower jaw and this part is the left half of the lower jaw and this part is right half of the lower jaw. Students, this picture shows the arrangement of different types of teeth in our mouth. Let us know them. This horizontal line indicates the partition between the upper jaw and the lower jaw, whereas this vertical line indicates the partition between the half of one jaw and the other half of the upper jaw. Let us see the arrangement of teeth in each half of the upper jaw. Starting from here, these two yellow colored teeth are incisors. These two are incisors and this green colored teeth is canine and these two pink colored teeth are premolars and these three blue colored teeth are molars. Similarly, if you see the right half of the upper jaw, the teeth are arranged in the similar manner. Two incisors followed by one canine, two premolars and three molars. And if you see the arrangement of teeth in the lower jaw, first of all in the left half of lower jaw, starting from two incisors, one canine, two premolars, three molars. The other side again two incisors, one canine, two premolars and three molars. Students, look at me. The arrangement of teeth: two incisors, one canine, two premolars and three molars. This half of the upper jaw. Similarly, the other side of the upper jaw: two incisors, one canine, two premolars followed by three molars. So, in the same way the teeth are arranged even in the lower jaw. Look at this picture, this clearly shows now what is dental formula. Dental formula explains the arrangement of four types of teeth in each half of the upper jaw and each half of the lower jaw. So, I represents incisors, C represents canines, PM represents premolars, M represents molars. So, the numerator values indicate the arrangement of teeth in the upper jaw and the denominator value indicates the arrangement of teeth in the lower jaw. Students, look at this. This table shows the type of teeth, their shape and their functions. Incisors, number of teeth are 8, the shape they are flat with sharp edges, that is chisel shaped and they are used in cutting and biting the food. Canines, the number of teeth are 4, they are sharp and pointed. Students, if you see in carnivorous animals, these canines in carnivore animals are well developed with long pointed edges, helps in tearing the flesh. Premolars, they are 8 in number, they are flat or blunt and rectangular in shape, they are used in grinding. Molars, they are 12 in number, they are also flat or blunt and rectangular in shape, they are also help in grinding. Both premolars and molars are used in thorough grinding of the food during the mastication process. Students, in the part 1, we understood the significance of mastication to properly digest the food. So, the structures which are present in the mouth are teeth. With the help of tongue helps in thorough mastication, while the salivary glands are stimulated, secrete saliva. The food is thoroughly mixed with the saliva, gets lubricated and moistened. Students, let us see activity 7, the action of saliva on floor. How does the enzyme salivary amylase in the saliva breaks down the large starch into smaller subunits, usually into sugars? Students, for doing this activity, the materials required are rice flour or starch solution, glass beaker with normal water, test tubes, a watch glass, iodine solution and a dropper. First, take a test tube, take a pinch of starch powder into the glass test tube, add some water to this test tube and shake it thoroughly to mix the water with the starch powder. Now, our starch solution is ready. Take some starch solution into the watch glass and with the help of dropper add few drops of iodine solution to this starch solution. So students, on the addition of few drops of iodine solution or tincture iodine immediately turns blue-black color, which indicates the presence of starch. Now, take this remaining starch solution into 2 test tubes in equal amounts. Do not add anything to this first test tube. Take the second test tube, add some saliva to this starch solution. So, I just added my saliva to this test tube. Wait for some time. So, after some time, I am adding again few drops of iodine solution to this first test tube. Which gave again blue-black color, confirming the presence of starch in the first test tube. Let us repeat the same activity by taking second test tube where we have added the saliva to the starch solution. I am adding again few drops of iodine to this second test tube. So students, observe carefully, this did not give blue-black color, which indicates the absence of starch in this test tube. Students, in the above activity, the salivary amylase, which is also called as ptyalin, converts the starch that is present in the food during the mastication process. The starch is converted into simple sugars. So, the presence of sugar is confirmed by doing the activity 7. Students, let us do activity 8, testing pH of mouth at intervals of 1 hour. What is the pH value of saliva? As you already know that the pH value of saliva is 6.8. Does the pH value of mouth changes? Yes. Whenever we take different food items, we thoroughly chew, and during the process of chewing the food, our salivary glands stimulate and secrete saliva that is thoroughly mixed with the food during mastication. So during this process of mastication, based on the amount of the saliva that is secreted and the type of food that is eaten by us, so the mouth pH changes. Let us know about this universal pH paper. So this is the universal pH paper with the different colors and different pH values. By using this paper, we can identify the pH of any chemical by dipping it in the chemical based on the color obtained. We can do this in our school laboratory by taking a small piece of pH strip. Before you consume food, place this piece of pH paper on the tongue. Match the color of this pH paper with the color chart and note down the pH value. So take at least 4 readings after consuming different food items and compare your values with that of your friends. Students, as you already know that the pH value equal to 7 is neutral, pH value below 7 is acidic, pH value beyond 7 is alkaline. You can do this activity after consuming food. After thorough mastication, the food becomes bolus, and during the process of swallowing, the bolus enters into esophagus through pharynx. Now, what happens to this bolus? Let us see by doing activity. Activity 9, making a model of esophagus to observe how bolus moves forward. Travel of food through esophagus. What kind of the tube is esophagus? How does mucus help in passage of food? Students, in the chapter Nutrition, Human Digestive System, you studied what kind of tube is esophagus. Let us see the structural and functional aspects of the tube esophagus. The structural aspects of esophagus. Esophagus is long tube, its upper end connects to the pharynx, whereas its lower end connects to the stomach. So, this is the long muscular straight tube that connects pharynx in the upper end and stomach in the lower end. It is a muscular and elastic tube. The functional aspects of esophagus. The inner walls of the esophagus contains a lining called mucus lining. The cells that are present along the walls of the esophagus secrete this slimy substance called mucus. The esophagus walls carry on a wave-like movement by contraction, which helps in passing the food from the pharynx to stomach. So, students, esophagus is a long straight muscular tube. It shows long wave-like contractions and relaxations with the help of smooth muscles, and these movements are called as peristaltic movements. These peristaltic movements helps the bolus to move forward from the pharynx into the stomach. The mucus line helps in the easy passage of bolus, as well as it also protects the walls of the esophagus against the hard particles that are present in the bolus. Let us know the role of esophagus in passage of bolus into the stomach by doing this activity. Observe how the movement of bolus takes place through esophagus. For doing this activity, the materials required are a waste cycle tube, 2 potatoes and oil to lubricate the potatoes and the inner walls of the cycle tube. Take oil, pour this oil along the inner walls of the cycle tube. Also add some oil on the potato. Now insert this potato into this cycle tube. Now students, observe carefully how the passage of potato occurs inside this cycle tube. I am squeezing the cycle tube such that the potato is moving along the walls of the cycle tube. Students, look at this picture. This is the esophagus, commonly called as food pipe, which shows a wave-like contractions and relaxations. So, these movements are called as peristaltic movements. So, if you see this picture, this is the bolus. Slowly the bolus is passing down and coming into the stomach. So, the wave-like, so see here, the wave-like movements are called as peristaltic movements. Students, what is peristalsis? Peristalsis is a series of contractions and relaxations shown by the smooth muscles that are present in the wall of the esophagus. A long wave-like movements that are generated in the esophagus by the smooth muscles are called peristaltic movements, which will propel the bolus or the contents in the digestive system. Peristalsis is an involuntary and important process in digestive system which moves food through the alimentary canal. Students, so the bolus reaches the stomach. Let us know the role of stomach in the process of digestion. Stomach, the mixer and digester. So, this is the stomach, a bag-like structure or a sac-like structure that is situated in the left upper part of our abdominal cavity. Students, why do you think the stomach is structured like a bag rather than a tube-like esophagus? In order to accommodate the food, the stomach is bag-like. So, it is not possible if the stomach is tube-like. It cannot accommodate or it cannot store the food. Peristalsis in stomach. The three major functions of the stomach are propulsion, grinding, retropulsion. First one is propulsion. If you look at this picture, these arrow marks indicate the movement of muscles that are present in the wall of the stomach. So, these movements propel the contents in the stomach from this part to this part. So, now this is called as propulsion. Here there is a valve called pyloric valve. This can be also called as pyrolic sphincter. So, during the process of digestion in the stomach, this pyloric valve is closed. It does not allow the contents to enter from the stomach into the small intestine. The second function of stomach is grinding. So, here the wall of the stomach contains muscles which show a series of contractions and relaxations. So, these movements are specifically called as churning movements, and these movements will help in grinding the food thoroughly inside the stomach. So, this is called as grinding. Even during the process of grinding, the pyloric valve is closed, restricting the entry of contents from the stomach into the small intestine. The third function is retropulsion. During the process of retropulsion, see, after the complete digestion in the stomach, what happens to this pyloric valve? This is relaxed. So, as it receives the signals from the brain, it relaxes a bit. During this time, the contents that are present in the stomach become soft slurry mass or porridge like material called chyme. Now, this relaxation of pyloric valve allows the chyme in minute quantities into the first part of the small intestine, which is called duodenum. Only small amounts of chyme is allowed to enter into the duodenum from the stomach. Immediately the pyloric valve gets closed and all these contents are reversed back into this stomach. So, students, we have seen the three major functions of the stomach: propulsion, grinding, retropulsion. So, by these functions the bolus has now become chyme. Now, this chyme is slowly entering into the first part of small intestine, that is duodenum. Students, look at this table. Food does not move uniformly throughout the digestive system. Students, the gastric glands which are present in the wall of the stomach secrete gastric juices. Of them, a strong acid is present called hydrochloric acid. The hydrochloric acid is strong enough to digest the hard bones as well. The HCl acid might do damage to our own stomach, but the stomach is not getting damaged by the action of HCl acid. How does the stomach protecting itself from its own acids? Let us know this by doing this activity. For doing this experiment, the materials required are 2 green leaves, dilute acid, dropper and Vaseline or petroleum jelly. Take the Vaseline, apply Vaseline on the surface of one of the green leaf. Leave the other green leaf free. Do not applying the Vaseline or petroleum jelly. Now, with the help of dropper, add few drops of dilute HCl acid to both the leaves. Leave it for about half an hour or so and observe the surfaces of 2 green leaves. Students, after 1 hour, observe these 2 leaves. So, this is the leaf where we applied the Vaseline. So, it did not get damaged by the action of dilute HCl acid due to the presence of this Vaseline lining. Whereas, the other leaf where we did not apply the Vaseline, so this leaf got damaged by the action of dilute HCl acid. Lab activity: how the stomach protected from the secretions of its own acids. To understand this, we will perform the following experiment. Which leaf was affected by the acid? What kind of change did you observe in the leaves? What saved the other leaf from the effect of acid? Students, after the completion of lab activity, the Vaseline applied leaf got protection from the action of dilute acid. The reason, the Vaseline, a layer protecting in our stomach, the mucus secreting cells secrete huge amount of mucus which forms a thick lining of the stomach. Now, this mucus lining protects the walls of the stomach from the action of HCl acid. Travel of food from the stomach to the intestine. Entry of chyme into the duodenum. The small intestine meanwhile receives the bile juice from the liver, pancreatic juice from the pancreas, and the intestinal glands that are present in the wall of small intestine also secrete intestinal juices. So, the chyme is thoroughly mixed with all these juices and its acidic nature is converted into alkaline medium. Let us see the shape of the small intestine. Students, look at this picture. The small intestine is highly coiled, a long tube. Students, as you already studied in the human digestive system, the function of small intestine is digestion and absorption of nutrients. So, once the digestion is completed, the absorption of nutrients takes place. The nutrients get diffused into the blood circulation passing through the wall of the intestine. Students, let us know the significance of villi by doing this activity 10. Now, take 20 into 20 centimeters chart paper, roll it and make it into tube like this. Now we prepared two paper tubes. One is of 10 into 20 centimeters size, another one is 20 into 20 centimeters size. Is it possible? No, it is not possible to insert this larger paper tube into smaller paper tube. Students, I have folded the larger paper tube into many folds. Now I am trying to insert this tube into this smaller paper tube. Students, see, now it is possible to insert this larger paper tube into this smaller paper tube. So how is this possible? By making larger paper tube into foldings. Now what is the relationship between finger-like structures and paper foldings? Now these foldings resemble the finger-like structures that are present in the wall of the intestine, which are called villi, and on the surface of these villi they contain microvilli which increase the area of absorption. Students, this is the schematic diagram of villus, the enteric nervous system. Enteric nervous system, the second brain. The brain in the gut confined within the wall of the gut. It contains more than 100 millions of neurons, that is more than the number of neurons that are present in the spinal cord. This enteric nervous system extends from esophagus to the internal anal sphincter. The enteric nervous system works independently to maintain the peristaltic movements along the length of the diastole tract starting from the esophagus till the internal anal sphincter. It is also involved in the secretory functions, though it is not involved in decision making, though it is not involved in conscious thinking. It is mainly concerned in taking decisions regarding the activities that occur in the diastole tract. So after the absorption of nutrients, the undigested unabsorbed food materials enters into the large intestine, where they are converted into solid fecal matter. Now let us see the expulsion of waste materials from the body to the outside. Expulsion of waste materials to the outside occurs in two major pathways. One is expulsion of waste via blood through the kidneys, skin, etc. Mainly the excretory substances are excess salts, water and the nitrogenous metabolic waste, which is urea. So this is nothing but the urinary system or the excretory system, where kidneys play an important role in the elimination of these substances in the form of urine. The undigested food materials and the unabsorbed food materials are converted into fecal matter and expelled to the outside in the form of stool through the anus. Students, look at this picture. At the opening of this anus, there are two sphincters. The one which is present on inner margin is called as internal anal sphincter, or on the outer margin is called external anal sphincter. As the signals come from the brain to this internal anal sphincter, these muscles relax, whereas the relaxation of the muscles that are present in the external anal sphincter are under our conscious effort. So these are voluntarily controlled. Students, so far we have studied how the digestive system is dependent on the other systems: the circulatory system, the respiratory system, the muscular system, the nervous system, the hormonal system. All these are involved in the process of digestion, absorption, elimination. What we have learned: types of teeth, their function, salivary amylase, the pH of your mouth, esophagus, its peristaltic movements, stomach, mixer and digester, stomach, its mucous lining, small intestine, functions of villi and microvilli, expulsion of waste, egestion through anus, anal sphincters and the energy releasing process. Students, for your assessment, you practice these questions at your home in improve your learning. Students, by this we conclude this coordination in life process. Let us meet in another session. Thank you.